Let's get into foundation models, and it's my pleasure to introduce our first speaker of today, Christian Schiffer from Forschungszentrum Jülich. Christian leads the Helmholtz AI research group at FZJ, and he works on AI methods for decoding the microstructural organization of the human brain. And today he will present his work on CytoNet, a foundation model for cytoarchitecture in the human cerebral cortex.
Yeah, thank you very much for the kind introduction. After the great keynote that we heard earlier, we are staying with biology, and I'm very excited to present you our most recent work on a foundation model for cytoarchitecture in the human cerebral cortex. So one of the core goals of our lab is to develop AI methods to describe human brain structure at micrometer resolution and at whole brain scale, including cells, fibers, and their function. For this, we acquire post-mortem human brains, we slice them into thin slices, process them, and then digitize them using high-throughput microscopic scanners for further analysis. This leaves us with different imaging modalities, including cell body stained imaging, allowing us to study the cellular architecture of the brain, polarized light imaging to study fiber architecture, and receptor autoradiography to study neurotransmitter distributions. Our goal with this work is to build a model, a foundation model, that captures these complementary architectural principles of the brain, encodes them, and makes them available for analysis, downstream tasks, and research. In this work that I'm going to present here today, we are going to first focus on cytoarchitecture to develop the methods. But it's important to note, to point out that the methods that I'm going to show you in a minute are also applicable to other modalities directly, and I even work to bring these modalities together, as you will see in a second. So what is cytoarchitecture? Cytoarchitecture is defined by how cells are and neuronal cells are organized in our brains. This includes the organization of cells into layers, as well as certain other properties like the shape, size, and general distribution of cells. Why is this interesting? Well, you all know that our brain has different areas which have different functions, and these different areas also tend to have different architectures. So essentially, by looking at this data, we are looking at a part of the hardware that is actually running these functions and helps us to understand it. So our goal, as I said, is to encode brain architecture to answer questions like what kind of cells are present, how are cells distributed, how are layers composed, which areas are formed, and also taking a look at the other modalities that I just showed, how are fibers organized and how do local networks function. And as I said, our approach here is to build a foundation model that captures these principles and makes them available. But the question is, how do we actually do this? So how do we train this thing? When we started this work, as one should, we had a look at what is already there, and what we did for the self-supervised training of this model, we had a look at SimClear, as a very popular method. And when we first tried this, we were very, very happy because this converged like textbook perfect. And it did not work at all on any relevant downstream tasks. So this was a bummer. And we asked ourselves, okay, how can this be? How do we debug this behavior? And to do this, we picked some example patches, as you see here from our data set. So some patches that we extract from our microscopy images, and you see them here along with their brain area, which gives us an idea of what cytoarchitecture they actually represent. And then using the features that we learned with our SimClear approach, we then queried what samples in our data set are actually similar to those, which we see here. And what we see here on first sight is that regarding the brain area that we see here, so regarding the cytoarchitecture, the cellular architecture, it doesn't match at all in most of the cases, which means that indeed it did not learn cytoarchitecture. But what did it learn? So with a little bit of imagination, you can see that in this case here, it seemed to have learned the rough shape of the tissue. And this seems to be the case in many, many cases. There are also other examples of this, but essentially here we have a case of shortcut learning where the model did indeed solve our self-supervised task without actually learning anything that is of use for us. And we had a look at, or thought about how can this come to be, and basically we found that this is a limitation of many of these methods which are based on this idea of learning from what I would call semantic consistency under multi-view augmentation, where we take an image, augment it in some way, and then assume that everything that we vary is not relevant, and we only keep the core relevant features that are interesting for us. There's also some theoretical foundation for this, which can be illustrated in this nice figure here from paper from Yonglong et al., where we basically see, briefly summarized, depending on how we choose our data augmentations for these learning methods, like SimClear, momentum contrast, Dino, Wallow twins, and others, we might end up in regimes where we are missing actually relevant information. So if our augmentation is too aggressive, for example, or we might have excess information if we fail to vary everything that is irrelevant, which is the case here. So we are basically failing to vary the shape of the tissue and so on, or the presence of blood vessels, which means that in this case we are unable to hit this sweet spot where we are exactly at the point of what we want to learn. So this ruled out immediately a lot of nice methods that we could have used. So we went back to the whiteboard and thought about, okay, how can we define a learning task that actually works with the data that we're looking at here? And one core insight that we had here is that when we look at our brain, it's not random, even though you might think that sometimes. It is organized in functional modules, which are localized and do certain things, and as I said, have similar architecture. Which means that if we tend to look at samples from similar locations in the brain, they tend to have similar architecture. And we thought, okay, that's a core principle, and we want to exploit this to define a contrastive learning objective for self-supervised learning. And to illustrate this, let's assume that we have two images. Here, two brain sections from two different brains, but from approximately the same location in the frontal lobe of the brain. We can now pick some samples, which are indicated here with these boxes, and we can see that even though they are in different brains, the orange and the blue one are quite close to each other, and giving our assumption that I just mentioned, we have to assume that they have similar architecture. So we want these to be similar in our model at the end of the day. While the purple one is quite far away, so we can assume that this has quite different architecture. So this is essentially the relationship that we want to exploit here to define our contrastive learning objective. Now we have a technical difficulty here, which is that we don't have a common coordinate frames for two brains. We can quite easily solve this by projecting our points into a 3D reference space, which is relatively easy. And now we have everything that we need for our framework. We can define distances in this reference space, and to train this, we can pick images from our microscopy scans. We can then pass them through a model. In our case, this is most often a ResNet or a hybrid between ResNet and Vision Transformer, giving us features. And now we go back to the distances that we compute in 3D space and derive the similarity relationship that we want for these vectors. We implement this with what we propose as spatial NCE loss. And this is the formulation here, which might look quite complicated, but if we take it apart, it's actually not that hard. The latter part here is the info NCE loss, which many of you might be familiar with, basically pulling together similar samples and pushing apart dissimilar samples. An important difference to many other methods is that here we don't only compare always two samples, but we do a pairwise comparison across the entire batch, because we can compute distances between any sample that we have. So why not also do that? And the most important difference is that instead of having a binary similarity relationship, like you have, for example, in SimClear, where you say either two samples are either similar or not similar, we derive a continuous notion of similarity, which is based on the Euclidean distance. So we took this, we trained models on a large data set that we have available at our institute, consisting of 10 brains, 4,500 sections, and about 11 terabytes, which is quite computationally expensive, also since we have to use quite large image patches to maintain the necessary field of view that we need for the tasks that we are doing. So after training this model, we wanted to look, okay, what does it learn this time? And for this, we computed features over all our data, over our entire data set, and then computed, among others, embeddings, and then looked at the results. And what did we expect here? So what we expected is that this actually clusters by brain area in some sense, by cytoarchitecture, because this is what we trained it to do. However, the result was this. This is a 2D UMAP embedding, colored by the brain the points correspond to, and you see that the UMAP space basically falls apart into brains, which is actually not what we trained it to do. So did we fail here? Did it not work at all? No, otherwise I wouldn't be showing this here probably. So we see that apparently the difference between individual subjects that we're looking at is actually larger in terms of cytoarchitecture than actually the cytoarchitectonic differences within one brain. But if we have a closer look, coloring these features by a brain atlas, giving us approximate information of brain areas, we see that even though we have brain-specific clusters, we can see that the internal organization is highly similar, and it's not only similar but also anatomically reasonable, which is nice. So this means that the features that we have here do indeed encode what we're looking for. And we were able to apply these features in a wide range of different applications, including classification of brain areas, segmentation of cortical layers, quantification of generative models, the discovery of new brain areas based on clustering, and also the prediction of certain interpretable features which are not as abstract as deep features. The question remains, what's next? I spoiled it already at the beginning of the talk. We are actually aiming for multimodal training here. So we want to include different imaging modalities, which is relatively easy with the method that I showed, because this distance similarity works across imaging modalities more or less directly. We also want to include text, which might be a little bit less straightforward. If you're interested in how this is going so far, please check out the posters of my colleagues today and tomorrow in the poster session. And finally, the next big step, as we already heard it on Fabian's slide this morning, scaling this up to multi-modalities and also to larger data sets will require more compute. So we are very happy to be one of the first pioneer projects which will be running on the new exascale supercomputer Jupiter, which is right now being installed in Jülich, which will provide the necessary computational budget which we will need. This brings me to the end of my talk. I would like to thank all my collaborators, colleagues, the funding agencies, and of course you for your attention. Thank you.
Thank you so much. And we have time for questions. Someone's getting up faster, so I don't get to ask a question.
Hi. Well, you may.
No, it's fine.
So really cool results. Can you relate that somehow to the problems that fMRI is having, where they're trying to sort of figure out what people are thinking by looking at MRI data, and they realize that this works quite well for one individual, but you cannot transfer what you learned in one individual to another? Does your data kind of reflect this, that the similarity between brains is larger than the similarity within one brain?
I would say it hints at it. So it is very, very hard to grasp. And actually, I have to mention that we don't really know yet what is the difference. So we know that there is a difference. And if you talk to a neuroanatomist, they will also confirm, okay, if I look at two different brains under the microscope, it is different, but it's very, very difficult to tickle out what is actually the difference. But this is actually very, very interesting, because now we have descriptors basically of the microarchitecture, which you can relate with, for example, functional measurements. Of course, rarely in the same subject, which makes this more complicated, but it's basically a tool that we are envisioning this to be used for. Yeah.
And that would bring me, if you allow me, to the second question.
I would give it, because there's someone queuing up there. I'm sorry, I would first start over there because we are running low on time.
Can I stay here and ask later? Can I stay here and ask the second question later?
I think we have time for only one more question, and this is why I would give it then. Sorry, you have to take it to the coffee break.
Hey, Matt, thanks for the talk. It was quite interesting. We are having similar problems, basically batch effects everywhere. So my questions were mainly the U-maps that you showed. Were those on the test samples, or were those like plotted on the training samples that were used in the architecture?
This was, it included the training samples, but so the pre-training samples, but it is only a very small portion of it. Okay. So it's actually interesting, I don't have the picture up here anymore, but there was one cluster which was actually held out from pre-training. So, and there you see that the internal structure is actually similar, but it looks like compressed, so it's less distinct in one of the axes, which kind of, I interpret this kind of as, by not including it in the pre-training, we we have difficulties getting like the very, very specifics of a specific brain, which is in practice for downstream applications not super bad, because we can always include this data in the pre-training process. But it's interesting that this also is reflected there.
Okay.
Okay.